My name is Rovi, and I'll be presenting on pathogen spillover. Here are the steps I took during my research. Firstly, I brainstormed. I remembered learning about this chronocyte hypothesis from a course last semester. The idea was that viral domains tend to mirror the domains of life. There was the presence of a fourth viral domain, which hinted at a corresponding domain that once existed. From that, I was interested in seeing how pathogens could be used to track metapopulations, but it was kind of a niche topic. So I moved on to brainstorming other pathogen-related topics in population ecology using ChatGPT. Pathogen spillover was the one that interested me the most. From there, I used Google and Wikipedia to find various scientific articles, and then Research Rabbit to build a timeline of studies. Pathogen spillover is a transmission of a pathogen from a reservoir species to a new host species. The pathogen must overcome genetic and physiological barriers, such as the immune defense of its host, to infect the new species. The reservoir population is the native host species of the pathogen, which will infect a new species host population. Zoonotic diseases are a specific type of spillover, where non-human species transmit pathogens to humans, which could occur in interactions as seen in Figure 1. Factors that influence spillover include distribution and abundance of the two species, contact rate, where pathogen transmission is more likely to occur with frequent contact between reservoir and host species, phylogenetic relatedness, where the more related the reservoir and host species are, the more similar their immune defenses are, therefore the less mutations required to overcome the defenses in host species. This is why we don't see viruses from a plant infecting humans. Pathogen mutation rate also plays a role, as those with a high mutation rate are able to adapt quickly against host-specific immune systems, allowing it to proliferate within the new species. Figure 1 shows a very close interaction between a monkey and a human, where pathogen spillover is at great risk due to their proximity and phylogenetic relatedness. When a pathogen enters a new host, there are multiple outcomes that could occur. The first one is a dead end. The pathogen will be transmitted to an individual member of the host species, but it will either die due to the host immune system or die with the host, which is what often happens in humans with rabies. Therefore, it's not likely to be transmitted to other members of the host species. The second outcome is limited transmission. The pathogen is transmissible within the host species. However, there's a limited spread, usually because of its high virulence and subsequently high mortality rate. The mode of transmission also plays a role, such as in the case of Nipah virus, which can only be transmitted through body fluids and is not airborne. Thirdly, there can be stable integration of the pathogen within the host species. The pathogen adapts well to the immune defenses of the host, and the host species becomes a new stable reservoir species. For example, the HIV-1 virus, which came from chimpanzees, has spread all over the global human population and has killed millions. Pathogen spillovers have occurred all throughout history. The Plague of Athens in 430 BC is one of the earliest documented cases of pathogen spillover. The exact pathogen of the disease is currently unknown. However, descriptions of the event point to a zoonotic disease, such as typhus or anthrax. In 1881, a Cuban physician named Carlos Finlay noticed a pattern of yellow fever cases with weather that correlated with mosquito activity. He theorized that yellow fever virus was carried by mosquitoes and spread to humans. He presented the mosquito vector theory in 1881 and was called the Mosquito Man due to his radical ideas. In 2004, Power and Mitchell provided the definition of pathogen spillover, where reservoir species with a high pathogen prevalence transmits them to host species. And now, in 2025, researchers are using AI models to predict reservoir species for viruses, such as orthopox viruses, like monkeypox. In a study by Cheng and their colleagues, they applied various models, including machine learning and predictors to forecast future reservoir hosts for the orthopox virus. I'll describe their study in detail later on. Here is a timeline of scientific articles related to pathogen spillover, which doesn't include the study in 1798. This was all generated on Research Rabbit. I'll go over a couple of studies, starting with an inquiry into the causes and effects of the variolae vaccinae, a disease discovered in some of the western counties of England, particularly Gloucestershire and known by the name of cowpox by Jenner in 1798. And yes, that was just the title. His objective was to see if cowpox gives immunity against smallpox. To test this, he found a dairymaid named Sarah Nelms who got cowpox from her cow named Blossom. He then inoculated his gardener's 8-year-old son with cowpox using lesions from the dairymaid. 2 months after, the boy was inoculated with smallpox to test if he gained immunity. The inoculation with cowpox gave the boy a mild fever and discomfort, which only lasted 10 days. When he was inoculated again, this time with smallpox, he didn't show any symptoms, therefore acquiring immunity to smallpox. The procedure was named vaccination, as vaccinia was a Latin word for cowpox. 200 years later, in 1990, Gojobori and their colleagues wanted to find the origin of HIV viruses, as it had not been determined yet at the time. To do this, they compared nucleotide sequences of HIVs and SIVs, short for simian immunodeficiency viruses, which come from monkeys and apes. They used 4 types of SIVs, for mandrills, African green monkeys, Pseudomangabes, and Rhesus macacox. They built phylogenetic trees based on neighbor joining, which links the closest relatives first based on genetic distance to minimize the total branch lengths of the tree. Figure 2 shows a phylogenetic tree of SIVs and HIVs based on the 3' LTR nucleotide region. The SIVs have an SIV prefix, and the rest are HIVs. The tree shows that both HIV groups and SIVs from African green monkeys and mandrills all diverged from one ancestor at a relatively similar time. Based on their phylogenetic trees, they concluded that HIVs are variations of SIVs and may have been a result of recombination between ancient SIV strains. The next study we'll discuss is by Greystock et al. In 2015. They were concerned that commercially produced honeybees caused pathogen spillover to several wild bumblebee species, causing population declines. They wanted to find if shared flower use contributed to the transmission of parasites between honey and bumblebees. To do so, they used 2 types of flowers, one bell-shaped and the other resembling a platform. 3 colonies of honeybees and bumblebees were each infected with parasites that the other species didn't have. The honeybees had Nosema apis and N. serene. Only N. serene is able to infect bumblebees. The bumblebees had A. bombi, C. bombi, and Nosema bombi. A. bombi is the only parasite that has been observed to affect bumblebees. They used 3 additional colonies for each species, which acted as potential vectors. They fed on shared and unshared flowers for 3 hours, and then were screened for parasites. The results showed that pathogen spillover occurred between honey and bumblebees. C. bombi was detected in honeybees, however it doesn't infect them, but it can retain viability if it was ingested and excreted out. N. serene was able to infect bumblebees as well, which has detrimental and lethal effect on them. The last study we'll discuss is by Cheng et al., published this year. Their objective was to find the effectiveness of predictive modeling on mammal and OPV compatibility, aka possible reservoir hosts around the world. A few of the reservoir species included red panda, mice, and cats. They used trait-based boosted regression trees, which are algorithm-based trees that improve accuracy by remaking trees based on the mistakes of the previous ones. The host ecological traits used included dispersal, offspring per year, and island dwelling, which is the proportion of the species in a genus that has at least 20% of their breeding range on islands. The viral genomic features used included replication and gene expression, structure and entry, host environments, host interactions, which are the ways that the virus manipulates host cells, and immune evasion, how the virus avoids being detected or destroyed by the host immune system. They found moderate to high accuracy of the predictive models. Using their models, they predicted host populations most at risk of OPV around the world. The top map shows the observed distribution of reservoir populations with OPV. The middle shows predicted reservoir populations of OPV, and the bottom one is susceptibility of human populations to OPV based on smallpox vaccination coverage. They found that areas with high OPV reservoir species diversity, such as Africa, had the lowest smallpox vaccination rates. Moving on to applications of the studies. The first one is to predict spillover events. Using predictive models like Chang et al.'s, predictions of spillovers should be made to prevent zoonotic pandemics, such as COVID-19. Knowledge of spillovers should also be informed to the general public, especially in places with great risk, to limit contact with reservoir species. Thirdly, vaccinations can be made from zoonotic viruses, such as the smallpox vaccine made from cowpox. In conclusion, pathogen spillover is the transmission of pathogens from one reservoir species to a new host species, as defined by Power and Mitchell. Spillovers have occurred throughout the centuries, with many historical accounts, such as the Plague of Athens and the bubonic plague. And current research involves machine learning AI to predict possible spillover events.